What's good, everybody? Welcome to a video on the current theory of aging. Now, this is primarily based on David Sinclair's current research and that kind of vein of research, but I wanted to make this video because I feel that he makes kind of a long-winded approach in his book and his podcast and his YouTube page that could really be summarized into, a few minutes long video. So I'm going to try and do that for you guys here and really focus on the biochemistry of what's happening and what he's talking about. So what he essentially says is that the aging process results from X differentiation resulting in senescent cells. Now, what does this mean? So initially you have a bunch of stem cells, and those stem cells, based on what parts of their DNA and ultimately proteins they express, they become a specialized cell, such as a sperm cell, red blood cell, a neuron, a skin cell, all sorts of different things. But it's important to remember that even though all of these cells have different functions and they express different proteins, they actually all contain the same DNA and the same chromosomes. They're merely expressing only certain parts of those of that DNA and those chromosomes, which make the specialized proteins that they use and ultimately result in their particular structure and function. So what's happening in X differentiation is when, differentiation is when you go from a stem cell to a specialized cell, whereas X differentiation is kind of going backwards. But instead of going backwards into a stem cell, these cells are actually going backwards into what are called senescent cells. They're kind of these zombie-like cells that no longer function properly and release inflammatory paracrine signals, which damage other cells in the body and kind of help them to turn into zombie cells as well that can wreak havoc. So how is X differentiation actually caused? One portion of X differentiation is the idea of the unspooling of different chromosomes. So normally your DNA is compacted, as you can see here. You have a DNA double helix, and this DNA is wrapped around proteins called histones, which help to compact them into coils and supercoils, and ultimately they're packed into chromosomes. Remember, we have 23 pairs of chromosomes, so we all have the same chromosomes, but what's important is that the DNA that is actually read to ultimately create different proteins, they're essentially told, each cell is told which DNA should be used based on different signals that they give to the histones. So most of the time, adding a methyl group to kind of open that DNA up so that the different transcription factors can come in and read the DNA and ultimately create mRNA and then protein down the line. So over time, due to various forms of DNA damage, instead of being wound tightly like this and only becoming unspooled in the areas that should be read, the chromosomes and DNA are damaged so that certain parts of the DNA that you don't want to be read are read. So, for example, in a red blood cell, instead of creating, red blood cell proteins such as hemoglobin, the DNA may become damaged so that the various proteins that make up hemoglobin are not created correctly. And ultimately, if you're not creating the protein structure correctly, then it can't function correctly. If this builds up into a variety of different cells, that can ultimately cause a lot of problems. So in addition to this kind of unspooling of the chromosomes that read different parts of the DNA inappropriately, this is also exacerbated by natural DNA mutations and telomere degradation that occurs. So DNA mutations, such as you can see here on the side, substitutions, insertions, and deletions. So substitution is like instead of having your normal DNA sequence, you have one base pair different, which ultimately creates a different amino acid. So you can see normally you have a G at this spot in the normal protein, which creates a glutamine amino acid. But in this case, it's substituted for an A, or an A instead of the G, which ultimately creates lysine. So if you have a different amino acid and within a protein that's created, that creates a different structure, and then the protein can't function properly. And this may seem a little complicated if you're having trouble kind of following along and you don't have a big kind of foundation in biochemistry or biology or genetics or anything. I'd highly recommend watching my Biochemistry Explained in 10 minutes. Or just, going and researching other videos on your own. It's not a very complicated thing, but I can understand if you just, jumped into it right now, how it'd be a little overwhelming. So in the same vein, you can insert. So there's a base pair that's accidentally put into the DNA strand. You get a different amino acid result and deletion. So you get rid of a DNA base pair, and then once again you have a different amino acid than originally intended. And this can happen due to a variety of reasons. Normal things such as if you're copying DNA every day for 80 years, there's just naturally going to be some errors that occur that can create these. Or you have DNA damage from the sun, or different compounds in the food you eat, or inflammation. There are a variety of things that can result in these different mutations that can exacerbate the chromosomal unspooling and ultimately X differentiation. The other thing that I wanted to mention was telomere degradation. And, I probably should have put kind of a diagram on here, but basically, when DNA is read, on the very ends of DNA, there are kind of just random base pairs that don't necessarily code for a particular protein, but they're there on the end of the DNA in order to protect it from becoming shorter and degrading. So that's what telomeres are. When DNA is shortened, instead of cutting off something important like this glutamine amino acid residue, you'll have a bunch of random base pairs on the end here that won't really code for anything, or won't code for anything important. And so it protects kind of the meat and potatoes of the DNA on the inside. And so, over time, those telomeres can degrade. And if they degrade to the point where they're not even there, then once again, you can have these kind of essential amino acids or essential base pairs in the inside be affected, which ultimately creates, a messed up protein. Last, the other thing that kind of contributes to the creation of these senescent cells due to the degradation of DNA and the unspooling of these chromosomes is mitochondrial dysfunction. So mitochondrial dysfunction can occur for a variety of reasons, but the reasons that it ultimately damages DNA is because of the drastic increase in reactive oxygen species. Now, I kind of talked about this a little bit in my immune system videos, so I have that same kind of organic chemistry reaction going on here, so you can kind of see what I'm talking about and how these reactive oxygen species can be damaging. So a reactive oxygen species is simply kind of a compound here that contains oxygen that has a lone electron that's really searching and really wants to pair with another electron so that it can be stabilized. So in this case, you have this hydroxyl radical, and it finds that electron so that it can be more stable in what is normally a relatively stable molecule in unsaturated fatty acid. So it takes the electron from there and the proton and the hydrogen, but now you have a molecule again that has basically, you've transferred this problem to the unsaturated fatty acid. So now that unsaturated fatty acid has a lone electron pair that really wants to be paired up, and another reactive oxygen species comes in to kind of fill that gap. So it appeases this carbon atom, but now you just have another, a brand new molecule with the same problem, and so on and so forth. And so by the time you're basically sending this problem down the line, and because of the interference in the natural structure of these molecules, ultimately it can cause damage because they aren't able to properly function. So how does mitochondrial dysfunction occur? It can occur from a variety of things. but the primary ways that this occurs is either through chronic stress or inactivity and overeating. So chronic stress creates these reactive oxygen species because it raises your cortisol and catecholamine levels, which these are kind of your stress hormones, so fight or flight. So what this does ultimately is it releases a lot of glucose into your bloodstream for your body to use, and because it thinks you're in a dangerous situation where you'll need a lot of energy. However, in this case, because you are, using all of this glucose in your electron transport chain of your mitochondria, like you're seeing here, naturally, just because the process is occurring so often, it's going to create all of these reactive oxygen species byproducts, which, once again, if you have all of that, all of those reactive oxygen species floating around in your cells, and they ultimately reach your nucleus and damage your DNA, they can contribute to that unspooling. The other way is through inactivity and overeating. So in the same way that those stress hormones, cortisol and catecholamine, will raise your blood glucose if you're stressed out, just overeating and creating, giving your body sugar and carbohydrates to increase your blood glucose levels will, without actually utilizing that glucose, will do the same thing. So then ultimately you can see down the line that the primary effects, basically you're just overloading your mitochondria and you're creating way too many reactive oxygen species, and those reactive oxygen species can wreak havoc, not only destroying your DNA in your cell, but also because you're kind of using that process so often, it can actually damage your mitochondria and make them less efficient with the glucose that you're using, because there's they're just getting so much in such a short period of time that they can't really keep up with it, and this causes issues. So ultimately, you see these downstream issues: telomere shortening, epigenetic dysregulation, and senescence. So that's exactly what we're talking about here. You have the DNA damage, the telomere shortening, which also damages the DNA so it can't be read appropriately, the epigenetic dysregulation, which is just a fancy word for talking about the specific DNA that needs to be read from the chromosomes in a particular kind of cell is not being read. It might accidentally read the wrong DNA portion, which will result in improper protein expression. And then you have, all of these things that are ultimately contribute to death and aging prematurely. So elevated blood pressure, cardiovascular disease, which is obviously the number one preventable killer, diabetes, neurodegeneration, physical and cognitive decline. So this last section here is just kind of giving an overview on what actually senescent cells are. So once again, these zombie cells release inflammatory paracrine signals which damage the body and create more zombie cells. So those paracrine is just a signal that is transmitted from one cell to the cells that are like nearby it. And those inflammatory signals are kind of cytokines, interleukins, chemokines. You can see that here. And if you haven't watched my immune system video, those are just molecules that signal the immune system to come and basically attack and kill certain cells. They're normally used to, kill bad cells in your body or harmful viruses or bacteria that have been there. But if you have this random zombie cell that is all of a sudden marking cells around it that are not actually detrimental to your body, and your immune system is killing those, that's where you get some of these autoimmune conditions or just inflammatory conditions like arthritis, for example. That's why arthritis is so common in old people. Some of the other things that you see in senescent cells, you can look up senescent associated secretory phenotype. It'll have, even more examples of what I'm talking about. But some things that happen are they develop a very unique surface markers they actually end up shedding their normal membrane-associated proteins and their normal signaling proteins, their extracellular matrix, and they create kind of a unique fibronectin surface. And so by changing their cellular membrane, they're actually able to become more motile and metastasize to different parts of the body. So then, even though they're not releasing these endocrine or system-wide molecules, inflammatory molecules, because they can move now and move throughout the body because of this new cellular membrane, they can go to other places and do the same thing. You also have the cell cycle arrest. So normally these senescent cells can be killed off by autophagy, but because of this new phenotype, this cell cycle arrest that's occurring, it kind of stays in the final stages and doesn't alert your body that it needs to be recycled. So normally you hear people talk about how cells have reached their Hayflick limit, which is just the idea that over about 50 cell divisions is about the lifespan of a single cell. Once they reach that, if they're not cleared out by autophagy, they'll stay as a senescent cell. This can also be exacerbated or occur more quickly due to mitochondrial deterioration, oxidative stress, DNA damage, chromatin disruption, oncogenes, things like that. Ultimately, if there are enough senescent cells and they're able to disrupt the DNA and the chromatin of other cells, that results in the aging and dysfunction that we talked about and those long-term tertiary outcomes that lead to death. So this became a little bit more long-winded than I wanted it to be. I tried to make it short, but I can assure you, even though it might be a little bit dense, it will save you from watching hours and hours of David Sinclair's videos and trying to research on your own because, at the end of the day, I really appreciate the work that he's trying to do. But at this point, he's just kind of trying to make money, and he's saying things in a very long form manner that if he really wanted to, he could just say in a very succinct manner. So I hope this was informative for you guys. I will be releasing some other videos after this to kind of talk about what you can do with this information and the particular pathways that can combat the creation of senescent cells. So thank you.